The overall goal of this protocol is to engineer artificial splicing factors that can specifically manipulate alternative splicing of a given target.
This method can help to answer key questions in the field of RNA processing, such as the regulation and misregulation of alternative splicing in cancer cells. The main advantage of this technique is its flexibility. We can design artificial factors that either promote or inhibit different types of alternative splicing in any given genes. Demonstrating this procedure will be Huan-Huan, a research associate of my laboratory, and Qianyun, postdocs in my laboratory.
Wild-type PUF DNA is used as a template for generating the customized PUF domain scaffold. Use PCR primers containing PUF sequences that specifically recognize different RNA nucleotides in each position. For each PUF repeat, use 4 different primers to recognize a different base on each position. In round 1 PCR, use the primer pairs shown to generate coding sequences for 4 PUF repeats, universal bridge fragments, and cap fragments. In round 2, use the PCR products generated in the last step as templates with the primer combinations shown to generate coding sequences for RNA recognition codes R1 to R4 and R5 to R8. In round 3, use the products from round 2 and bridge 4 to 5 as templates with the R1 forward primer and R8 reverse primer to generate coding sequences for R1 to R8 without caps. In round 4 PCR, use R1 to R8, 5' end cap and 3' end cap as templates to generate coding sequences for